Major molecules of the cell. The four major molecules of the cell are carbohydrates, lipids, proteins, and nucleic acids. The first category, carbohydrates, are a ready, easily consumed cellular energy source, and most cells only can consume monosaccharides. Monosaccharides, simple sugars, include glucose, fructose, and galactose. Disaccharides are covalently bonded monosaccharides. Sucrose is glucose and fructose. Maltose is glucose and glucose. Galactose and glucose, I said in the last set of slides, is a milk sugar. That's lactose. Polysaccharides are lots of these monos branched together, more than 3. Energy is readily stored as polysaccharides. I had also mentioned that we have a molecule called glucagon in our bodies. You bring in calories from whatever source. Your body really wants it to go to energy first, all other things being equal. So it's going to take whatever you consume, fats, proteins, whatever, sugars, and break it all down into simple sugars. Some of this is a complicated process, and some of it's quite simple. If you consume a bunch of lactose, it can break it down, pop off the glucose from the galactose with an enzyme, and voila, you have your simple sugars. Other things like breaking down amino acids are a lot more cumbersome, but it can happen. So your body tries to turn, again, all other things being equal, tries to turn everything into an energy source of monosaccharides. If your monosaccharides don't get consumed for energy, your body's going to want to pack it away for a rainy day. Our long-term storage of energy is fat, right? It's the fat on our bodies. We do have an intermediate place though, and that's glucagon. Your blood, the free glucose that's left over that your cells aren't really keeping up with or catching up to, goes through your liver. Your liver takes it and packs those molecules down and covalently bonds them into these branching, fairly dense sugar chains. This is a really good semi-long-term energy storage source. It's relatively easily broken down. It's pretty available close to the bloodstream. You don't need to do any special cells or tricks or anything to make this happen. When that sits around in your liver for a couple days and hasn't gotten used, then it can be packed on as fat. At any given time, you store about 2,000 calories worth of glucagon in your body. Carbohydrates aren't just used as an energy source, though. They can also be structural. We're going to talk about this more in the cellular unit, but we find a lot of polysaccharides as markers on the outside of the cell that can act as either receptors or can help tether it to other cells or can identify the cell as being self or non-self so that your immune system knows whether to attack it or not. Lipids are triglycerides. Triglycerides are a hydrophilic head with multiple hydrophobic carbon chain tails. You find this as phospholipids overwhelmingly in the body, where you have this hydrophilic head that contains a phosphate group right here. Okay? Remember, what are our hydrophilic or electronegative molecules? We have nitrogen, we have chlorine, we have oxygen. Phosphate is another one too. So you got a bunch of oxygens here, you have this nitrogen here. Those are all hydrophilic things. You got a couple oxygens right here, but overwhelmingly the bulk of this molecule is carbons, which are not electronegative. We didn't mention that as being an electronegative molecule, and it's studded completely over with hydrogens. Remember, carbon has 4 electrons in its valence shell. Its atomic number is 6. You have 2 electrons in the first shell and 4 in the outermost shell. That means at any given time, it can happily, easily, readily make 4 bonds with its neighbors. It's very enthusiastic bonder. It's part of the reason why it's such a great molecule to be a basis for life, is it's so inclined to build things. So what you have with your fatty acids in your body overwhelmingly is this philic polar head and phobic nonpolar tail. A steroid is basically the same thing, but with a ring formation instead of a chain. And a fatty acid has a glycerol head instead of a phospholipid head. Fatty acids also have 3 tails. Phospholipids have 2 tails. This part here, the long phobic stuff, is your tail. These, when you have your carbon chain tail, so if you just take a look at this right here. That's your carbon chain tail covered in hydrogens. So you look, and every single carbon is making 4 bonds. The next one, 1, 2, 3, 4. The next one, 1, 2, 3, 4. Every carbon in that line is making 4 bonds. That's the most it can make to be stable. It's completely covered. Therefore, we call it saturated. The carbon chain is saturated with hydrogens. That's a saturated fat. When a carbon chain is saturated with hydrogens, it lines itself up in this nice straight line. Fats that are heavy in saturated fats tend to be solid at room temperature. Butter, for example, lard, a lot of animal fats, frankly. Animals like to pack on their fat as saturated fats because it's just super dense. Think about this straight shape and how, because it's straight, there's no kinks or curls or anything to it, you can really pack in those molecules very closely to one another. Pack them in like cordwood, kind of. So you can store a lot of molecules inside of any given fat cell, and it's a very efficient use of space. If you have a carbon that does not is not completely covered in hydrogens, it's still going to want to make 4 bonds. So what it'll do is it'll double bond with the carbon next to it. That double bond is a little extra tight compared to the bonds that carbons and hydrogens make. And as such, when it pulls in that extra a little bit, it bends it. That bend means that it packs less densely. You can't stack those molecules up quite as nicely as you can a saturated fat. Those are called unsaturated fats, and these tend to be liquid at room temperature. They're oils. They can be less densely packed, therefore they're disinclined to be solid. These are extremely valuable energy source for the body. When you oxidize these molecules, you're going to release, when you give away electrons, because there's a boatload of electrons in these big old molecules, right? And the way it's configured, and I'm not going to go into the details, makes it sort of amenable to giving up these electrons. When it does that, it releases tons and tons and tons of energy relative to something similar. And when you release energy into the cell can use that, provided something's close enough to the reaction happening in the first place to make use of it. These are very efficient energy storage molecules. One more thing I want to mention is I get a frequent question in lecture when I do this in person about trans fats. What that basically is you have a different direction of bonding going on, where you have molecules, instead of something sitting out like this in the molecule, they'll sit kind of opposite. And it refers to the orientation of hydrogens along that chain. That's something that you don't really find so much in nature, but when we try to turn unsaturated fats into saturated fats in a lab or in a factory, that's what you end up with, is this kind of functionally saturated but kind of wonky molecular arrangement that makes unsaturated fats solid at room temperature. They act like a saturated fat. So margarine, for example, is vegetable oil, which is liquid at room temperature, but it's been modified in such a way, it's been just like blasted with hydrogens so that it takes on saturated properties. But since the molecular arrangement isn't exactly the same, our bodies don't use it in the exact same way. And there's cardiovascular implications behind that. Proteins are the next major macromolecule of life, the building blocks of which are amino acids. This whole table over here is all of your different amino acids. There's 20. Amino acids are the building blocks of all proteins. There's some unique, some common features to all amino acids and some different features to all amino acids. The backbone of amino acids is the same. This top part that's not highlighted on all the molecules, you'll all notice they start with H3N plus and then COO minus. It's exactly the same. Take note of the plus and the minus. That's going to come in handy in a bit. The highlighted part down below is what makes each amino acid different from one another. This CH2 with one ring is what, on the same backbone as everything else, is what makes phenylalanine phenylalanine. Add another ring to it in a nitrogen group, now it's tryptophan. Take away the rings, but add a sulfur and a couple more carbon CH3s. Now it's methionine. The backbone's the same. The tail part down there is different. The tail part that's different for every amino acid is called the R group. For exam purposes, and whenever we're speaking with technical language, you're going to always want to refer to it as the R group. But when we're just hammering out concepts and still learning, it's okay to, colloquially refer to it as a tail. Some of those R groups are more hydrophobic, and some of them are more hydrophilic. And whether they're hydrophilic or hydrophobic will determine how it can bond and what shape the overall protein will make. Polar things tend to be attracted to other polar things because they have something in common. And non-polar things don't have anything in common with polar things like water or other polar amino acids, but they do have something in common with one another. Non-polar and non-polar have things in common, and so they'll end up not being attracted necessarily, but they'll end up being glommed on to one another by virtue of having nothing to do with all of the polar surroundings. When amino acids bond to make a chain, to make a protein chain, what you're going to do is a trick of dehydration synthesis. So if you take note, that backbone, let's take glycine and alanine. You have O minus in the background, in the one end of the backbone of the amino acid, and an H3N plus. That minus on the oxygen means it has an extra electron. The plus on the nitrogen means it has one too few electrons. It's positively charged. So what's going to happen is an enzyme is going to come in, strip away two of these H's and this O. Now what you'll have, so we don't have this, we don't have this. Now what we have is an oxygen hanging out here, a carbon that's only making 3 bonds now, a nitrogen and only 1 hydrogen. You're going to be able to make, this carbon wants to make an additional bond, so it's going to make a covalent bond because they're next to each other. You stripped out a water. Now you have these naked ends that want to bond with one another. They're next to each other, so they're going to bond with one another, covalent bond. The backbone of these proteins being covalently bonded and being very repetitive is very, 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 very strong. The R groups do not bind to one another, but by virtue of being next to each other and either having or not having polarity, they might be attracted to one another. Again, they're not going to bond exactly, but they might want to bend in a little closer to one another, or not. When you build a protein in the first place, you just string along a bunch of amino acids using that dehydration synthesis trick I just showed you. And you end up with a sort of string of pearls line kind of a configuration. Now, when you have an amino acid that's polar, remember all of this is happening in the aqueous environment. The cell, water is polar. Next to another polar amino acid, and these are the R groups. There's your backbone. This is your, I'm going to do this a different color just so it's easier. So the backbone is red, and the R groups are green. Polar is green, non-polar is blue. When you have two polar amino acid tails, R groups, next to each other, they're going to want to interact with the waters around them. These tend to splay out the backbone. So they end up kind of pointing outwards a little bit because they just want to interact with everything around them. So they spread out. When you have two nonpolar amino acids next to each other, they don't have anything to do with the polar environment of the cell. They have more in common with one another than the water around them. And so that backbone tends to bend in. The molecules of the backbone are Loctite, ironclad, bound to one another, but that does not mean that they are a rigid straight line. They can still bend. So these polar molecules are going to be inclined to kind of glom up with one another because they don't have anything to do with the water around them, and it'll create a different type of bend in the backbone. When you have these types of bends, that's going to contribute to the secondary structure of the cell. The first line of amino acids to make a single string protein chain is your primary structure. Different R groups of the amino acids are going to either attract to the water of the cell or not attract to the water of the cell, and will create a secondary structure. Those attractions or not attractions happen spontaneously. There's no intervention here. No enzymes, no nothing. It's just based on charges. So that chain can then spontaneously fold onto itself to create the secondary structure. So this is really just the primary structure, but in like a zigzag or a curl. The curls are called alpha helices, alpha helix singular, and the zigzaggy pleated sheet, it's called, is the beta sheet. Those will then glom up again, because now you have a new arrangement of these molecules. You also might have intervention by an enzyme to help fold it in a little bit. This three-dimensional shape that the protein takes on, where it's not just an alpha helix or a beta sheet, but it's a series of these weaving in and out of three-dimensional space, is the tertiary structure. Primary first, secondary second, tertiary third. Finally, the complete chain in a completely folded 3D form, and then linked to other folded peptide chains in their tertiary structure forms, is a quaternary structure. This is multiple tertiary structures. Could just be two, could be a lot. Hemoglobin is a classic example of a quaternary structure protein. This is 4 smaller proteins that are bound up together. Each one has its own little spot with an iron in the middle. That iron molecule attracts oxygens. And you have 4 of those proteins mashed up together and bonded by enzymes with the help of enzymes so that you can carry more oxygen in a smaller packed space than you could otherwise. The 2 major ways that you find amino acids, or proteins rather, in the cell are in a fibrous form or a globular form. Fibrous proteins are insoluble in water and they're quite stable. They tend to be more saturated with hydrophobic molecules. Their shape is long and narrow. They tend to have a structural function in the cell. I'm just looking at this table here in pink. The amino acid sequence tends to be repetitive, the same few over and over and over and over and over, repeating again. By this repeating over and over, you're going to force this into smaller little secondary and tertiary structures that are going to arrange themselves fairly spontaneously into a line. They tend to be less sensitive to changes in pH and temperature, etc., by virtue of being overall structurally stable and repetitive and kind of mostly attracted to itself. It's not going to be easily tweaked by changing the environment. Examples of fibrous proteins are collagen. You've heard of collagen. That's that sort of protein that you find all over your body in a lot of different places. It helps attach stuff to other stuff. Myosin, you find in muscles. Fibrin, actin, keratin, elastin. A lot of these types of proteins, that usually refers to proteins. But these ones that you hear and see in a lot of like cosmetic products and shampoos and stuff are generally going to be fibrous. Globular proteins are more functional. These are your proteins that are going around and doing stuff. So think of fibrous as builders and globulars as doing stuff. When you close your eyes and picture an enzyme or picture a protein doing stuff in the cell, does it look like a string or a ball? Right? Every illustration that you get shown shows it in like this little globby ball. Globular proteins do stuff. These are made of proteins arranged in at least a tertiary structure, often quaternary. Fibrous doesn't necessarily need that. Fibrous could just be attracted to itself at the secondary in a secondary structure and then not really have anywhere else to go from there. It's so self-contained and stable. Globular requires at least a tertiary structure. They tend to be water soluble, therefore they tend to have a lot of polar amino acid tails, R groups. These are chemically active. Your enzymes are overwhelmingly globular proteins. You also see them listed in some literature as functional proteins, but for my purposes for the exams and for A&P 2, frankly, purposes, you're going to need to refer to them as globular proteins. Globular proteins tend to be round and spherical. They have a functional purpose. Their amino acid sequence is irregular, so you've got a lot of different things going on. You don't have this regular repeating rhythm that's inclined to create long strands. Instead, it's going to glom up on itself a little bit more randomly. They are more sensitive to changes in pH and temperature, etc., partly because they have more polar groups on them, more polar R groups. Examples are all of your enzymes. Hemoglobin has a quaternary structure. That's what you find in red blood cells to carry oxygen around. Insulin is the key that unlocks your cells to let glucose into the cells. Immunoglobulin, you find that in your immune system. And again, these are generally soluble in water, again, as a virtue of being rich in polar R groups. Finally, DNA and RNA, nucleotides, nucleic acids, are our last major macromolecule of life. Your four major macromolecules are adenosine, abbreviated as A, thymine, abbreviated as T, guanine, abbreviated as G, and cytosine, abbreviated as C. These are all in DNA. RNA has all the same ones except a tweak. RNA, instead of thymine, it has uracil, abbreviated as U. You just swap out, if you want to change a DNA code into an RNA code, you just swap out the T's for U's and there you have it. As I had mentioned previously in our hydrogen bonding conversation, nucleotides are bound to the backbone. See, here's your backbone by covalent bonds. Here's another representation. So this is your backbone, your sugar phosphate backbone, and this is your nucleotide over here. You notice between them, there's a solid line that binds them. Those are covalent bonds. Covalent bonds are the strongest ones we have in an aqueous environment. You're not breaking those without interference from an enzyme or like a blast of UV radiation from space. The nucleotides are not bound to other nucleotides quite so tightly. You do need to unzip DNA sometimes to like replicate it or to transcribe it or something like that, right? You need to do stuff with the DNA, so you have to be able to unzip it. You don't want it to be unzipped too easily, but you do need it to be possible. Hydrogen bonds, if you recall, are individually quite weak, but collectively very strong. Floating around in the aqueous environment of the cell, that whole long, well, it's not floating around, but you get the drift. That whole long length of DNA, 6 feet of DNA in every single cell, is zipped down the middle with a series of hydrogen bonds. All together, that's very, very strong, and it keeps the two sides of the two backbones of the DNA strand sealed unless something specifically comes in to break it. You can think about the zipping of the hydrogen bonds of a DNA strand like the zipper of your jeans. If you look at your jeans, you have these little, like, I don't know what to call them. I don't know what zippers terminology is, but you have like this claw-shaped side, another claw-shaped side. And when you zip it up, they mesh in with one another, right? Now, if you only had one of those little tongs or tines or whatever you call it, touching another one, it's not going to hold your jeans together. You just have one of them there. You can break that, no problem. But when you have all of them locked in together in a line, it takes a fair bit to bust open that zipper, right? The main differences between DNA and RNA. DNA is double-stranded. RNA is single-stranded. RNA also has an extra oxygen in its backbone. Deoxyribonucleic acid is DNA, and ribonucleic acid is RNA. You notice that RNA is lacking the deoxy part, D meaning lacking and oxy meaning oxygen. So ribonucleic acid, RNA, has the same backbone as DNA, but an extra oxygen. It also uses uracil instead of thymine. DNA ends up spontaneously curling into that classic double helix shape by virtue of molecular attractions that we'll get into another day. All of your heritable genetic information is conserved in your genetic code in DNA. Everything you got from mom and dad, your entire human operating manual that your body relies on to make every single cell work, that manual is DNA. You have some DNA in the nucleus of your cell. You also actually have a little bit in your mitochondria. Way back in evolutionary time, you had alpha proteobacteria. You still have them, but back then. And you have these special cells that are sort of eukaryotic-ish, but not quite called archaea. At some point, it's hypothesized that the archaeon consumed a mitochondrion. And the relationship proved to be favorable. The alpha proteobacteria could make energy that the archaeon couldn't, and the archaeon, by having engulfed that alpha proteobacteria, was keeping it safe. Seemed like a happy symbiotic relationship that eventually evolved into a dependent relationship. The alpha proteobacteria lost a lot of genes where it could live outside of the cell. The archaeon lost a lot of genes that allowed it to make any energy of it by itself at all. And they became a eukaryotic cell. You do still retain some DNA in your mitochondrion though. So it's not like the alpha proteobacteria just lost all of its DNA. It still has some, and it's unique to itself. And it looks a lot like bacterial DNA, which is pretty cool. All of your mitochondrial DNA, big fat asterisk here because there's a little bit of an exception that we're just now starting to learn about. All of your mitochondrial DNA comes from your mother's side. Your DNA from your nucleus come equally from both your mother and your father. The expression of your DNA might be tweaked more in one direction from another, but just in terms of bulk, chromosomal bulk, you have the same amount from your mom and your dad. RNA doesn't really encode, it doesn't create that master manual for the whole body, but it does do the work the DNA tells it to do. DNA is a big, giant tome in the middle of your nucleus. It has all the instructions for every cell in your whole body in every single cell. Your skin cell has skin DNA and hair DNA and eye DNA and kidney DNA and foot DNA and whatever DNA. It has all of it, and it just turns on what it needs. Now pretend you have a huge operating manual in your basement that tells you how every single thing in your town works. It tells you where everything is, who lives there, how every single house is wired, how many shingles is on every single roof. It tells you about that, about your house and every other house in your town. If you need to replace a shingle on your roof, do you want to drag that whole manual out up to the roof and hunt through it? Or do you want to go to the basement, look for the instructions for replacing a shingle, just copy that part and just bring that little bit of instruction to the roof? You just want to copy it and bring that little bit up to the roof. Your DNA in your nucleus represents that huge manual for everything in the whole town. Your body has a process to go through when it wants to make something or do something. It wants to bring those instructions into the cell, but you don't want to bring the whole manual out. So it goes in, it makes a little copy of what it needs, and brings that copy out into the cell to do stuff. That little copy of a special small section of DNA is RNA. Messenger RNA is what you're really sending out there. There's other kinds of RNAs too, but ultimately they're smaller molecules, single-stranded, that are based off of information from your DNA. They're created using information from your DNA. And they go forth in the cell and they do stuff. Your nuclear DNA, as best as it can, stays inside the nucleus of your cells.